Okay, to start our cross-program presentations, we've got Mike Steele from Canterbury Museum. He's going to tell us a little bit about phylogenetics.
Cheers, thanks. Okay, so this is going to be very much a tag team effort. I'm going to start off just by giving a little bit of background to what phylogenetics is about, and then Vince is going to talk about some recent work on what's called phylogenetic diversity. That's something we're currently studying from different angles, and then Daniel's going to talk about what are called phylogenetic networks. Let's see if I can get this to work. Okay, so I thought I might just start with some general comments about the relationship between, maths, physics and biology, and we all know that somehow maths and physics are great bedfellows. They work very well together. In fact, the comment's often been made that they seem to work almost too well together, that maths seems to be almost unreasonably effective in studying problems in physics. But the situation with biology is perhaps a little less impressive, or it was at least in the 80s. This delightful comment by Juan Carlo Rota in 1986: the lack of real contact between mathematics and biology is either a tragedy, a scandal, or a challenge, and it is hard to decide. He was a mathematician, yeah. So clearly, the interactions between math and biology are perhaps not quite as clean and clear as they are between maths and physics. But I guess we're trying to do something to rectify this sorry state of affairs. Okay, so phylogenetics is really the study of evolutionary relationships in a very general setting, and mostly that's in the context of biology. The idea that you have a common origin for all species, that they descended according to some sort of process of evolution, that you have speciation and extinction. You have some species in the past perhaps that becomes separated into different subpopulations, and they eventually become sufficiently differentiated that they really become reproductively isolated and form their own lineages that then speciate further in the future. And of course, this idea and the mechanisms by which this might occur go back to Charles Darwin, and this is the one of his, I think, his first sketch of an evolutionary tree in one of his notebooks, and there's some further, slightly more embellished illustrations of how he thought perhaps mammals and kangaroos and so on might have been related. Incidentally, as I go along, please feel free to interrupt and ask any questions. We have lots of time, and we've tried not to pack too many slides in.
So in Darwin's picture, there's one line can branch into 2 or 3 or 4.
Yes, that's right. There's this.
Is that the limit, or is it?
Well, biologists do often have these things that are called polytomies, and there's this idea of are we really saying that at some moment in time there was this, simultaneous radiation, or is it oftentimes for biologists this is really just some statement that we're not really sure the correct order of speciation, but we just represent it as a radiation from a point. I guess the usual assumption is that speciation is a binary process, that it's unlikely somehow that 3 things simultaneously form.
So this really becomes a black box where each of these lines quite.
Yeah, exactly. So soon after that, of course, there were these wonderful pictures of if you like, the tree of life. This is Ernst Haeckel, one of his, he drew many of these pictures, and you see here the way they viewed life there. You had 3 major branches. You had the animals, and you had the plants, and you had everything else, basically, the bugs, if you like. And that picture persisted for quite a long time until, I guess, biologists started to look at ribosomal RNA, and they found quite a different picture of life. In this picture, this is from the Olson and Woest, the plants and the animals somehow get relegated out to a tiny little corner of the tree. Here's fungi, and there's 3 main groups. There's eukaryotes, and then there's the bugs and bugs. So there's still 3 groups. It's just now 2 of them are bugs. To be more precise, you have the bacteria, the eubacteria. These are the things we usually think of as bacteria, E. coli and so on. But you have the second big group, very diverse and very different to these other bacteria, called the archaebacteria. And these live in very often in very hostile environments, perhaps in volcanic vents and so on, or very sulfur-rich environments. And genetically, they're as different to these things as these are. So there's really quite a different picture emerging here of the tree of life.
To characterize in this case?
Right, so this molecule is found in all of these organisms. So you can compare this molecule and you can say, how different is it between them? You know, how much does the sequences line up? And you get a measure of how much genetic change there's been over time. That's the idea.
So this picture is based on RNA, not DNA?
RNA, exactly. I think it's 16S ribosomal RNA. Yep. You need to find things that are present in all of these. Some of them don't have nuclei. Some of them, they're very diverse taxa. And then, the genetics area, of course, you can point to many individuals that in many places where there were fundamental advances. Certainly, in the late '60s, 2 very important things that happened were the realization that protein sequences, the sequences of amino acids, you could use those to reconstruct the history of, in this case, vertebrates, and see that they agreed very well with the fossil record. So that told biologists at the time that the information in the protein sequences of present-day individuals carries with it some sort of historical record of their evolution. And this was quite an important step. Then Anthony Edwards, who's perhaps well known to some of you here at Cambridge, was at Gonville and Keys College. He worked with Cavalli Sforza in developing mathematical methods to analyze gene frequency data, blood types and so on, to infer ancestry of populations. So these were 2 of, a handful of important developments in the '60s. And since then, there's been a real acceleration in this field. This is a little graph that Mark Pagel put together. He just used Science Citation Index and typed in the words molecular and phylogenetic and said, well, find me all papers that have those 2 words in the abstract, and just drew this graph. So you see that the use of molecular data for inferring phylogenetic relationships has certainly taken off. Certainly started taking off quite dramatically in the 90s. And coupled with this, and one of the reasons for this development has been the enormous increase in the amount of genetic data that's available in different gene banks. So in the early days, in the 70s, people were slowly sequencing these protein and later DNA sequences. But techniques got better. New advances such as PCR suddenly allowed for much more rapid sequencing, and now we have these genomic projects, and the amount of data is quite astounding. So what's phylogenetics used for? I thought I'd just spend a few minutes perhaps giving a little bit of an overview of the ways in which people use phylogenetic techniques. First of all, it's very important in evolutionary biology for determining the relationship between different species. So I'm going to have to do this manually. For example, here's some primates, and you want to know, well, how are they related, and maybe when did they diverge from each other? You might want to study processes of speciation. What does the shape of this tree tell us about speciation? Were there rapid bursts of speciation and extinction and so on? We may want to use this tree and the genetic data to understand better how DNA evolves, how proteins change, processes like selection and so on. It's also very useful in ecology. You find some new beastie out in the jungle somewhere, you have no idea what it is. Is it a is it an amphibian or something else? So you sequence it and you apply a phylogenetic analysis. You see where in the tree of comparable species does it fit, and you say, it's one of those. It allows you to somehow identify species, and this can be very important for, say, a new bacterial disease. You want to know what's it related to. It's very useful in biodiversity. This is something that Vince will talk about. Co-phylogeny, this is the study of how hosts and parasites co-evolve. And the migration of populations. So far we've been speaking, if you like, of different species, but you might just have a single species, like, for example, the human population. And a good example of this is the famous out of Africa hypothesis. I guess until the early 1980s, it was generally believed that all modern humans somehow evolved all over the world to their present state. But in 1986, Alan Wilson and his colleagues took mitochondrial DNA that's passed from mother to daughter, and were able to, they sampled mitochondrial DNA from, I think, well over 100 women around the world, and they were able to reconstruct a tree, and to date, when was the origin of this population? They got a very surprisingly short date, and since then the current theory is that all modern humans are in some way descended from a migration, or maybe several migrations, out of Africa approximately 200,000 years ago. There's much debate about the exact timing and so on. And that's really revolutionized, I guess, the way anthropologists think about the world.
Didn't they also argue that there was a single woman?
Well, that's a lot more debatable, I think. I'm not so sure they argued for it, but some people tried to argue for this. That sounds, whenever one deals with human evolution, you get all sorts of interesting perspectives. Certainly people have looked at the question of what was the likely founding size of this population. It certainly was, it wasn't one or two. It was probably in the order of thousands rather than a small number.
Like the African Eve hypothesis just mentioned, wasn't that really the statement that the mitochondrial DNA that we see today all comes from one person? Because you probably had a number of women all existing.
Oh, sure. But you have this bottleneck and you have this anomaly.
Yeah, and then the other one just got lost.
Yeah.
The other
But
sequences.
that still does mean that they all came from one woman.
Oh, the sequences did, yeah.
They came from one individual, might
One
not have been a
individual.
woman.
Sorry?
They came from one individual.
So it's women.
One female individual.
Mitochondria are matrilinearly.
Yes, one female, but it might not be coexistent with what you define as human.
But it's, but the other women that were around there may have contributed to the gene pool of today. It's just that this particular molecule, there may have been one woman indeed. It's more likely, even in this case, it was more than one. From
what?
It's likely in this case that it was more than one, even for the mitochondrial study here. But that's still a little debatable, I guess. Yeah.
Isn't it sort of very easy to distinguish between sort of a one-headed tree and a two-headed tree? Big difference.
In theory, it is, yeah. But of course, this tree is almost certainly not the true tree at all. It's just some estimate of the true tree based on a particular method for reconstructing trees from sequences. So
presumably in the last 20 years this has been redone, for example.
yeah, I think the short answer to your question is there's simply not enough information in the data to be sure, because these sequences are relatively short in comparison to the sort of questions that are being asked. I mean, there's, I guess, some robust conclusions one can draw about timing, but trying to say that it was specifically where this individual lived or whether there was more than one, it seems to be quite hard. But certainly I mean, you can model the ancestry of a gene back through time. This is the coalescent process that John Kingman, who is the former head of the Newton Institute here, did a lot of work on in the 70s. And if you apply that model, it would predict that there would be around about one, you'd expect perhaps one. However, the assumptions of that model are very, very special. You have a constant population size, and everyone's mating randomly, and it's quite hard to extend that rigorously because there's so much unknown about the population migration, I guess. Epidemiology, there's a lot of work in applying phylogenetic methods to viruses like HIV and influenza to try and understand how different strains are related when they developed, where they might be going in the future, I guess, and perhaps for designing vaccines. Some of the techniques from biology are used in other areas as well. They're used in stematology, which is the study of how in the days before printing presses and so on, different versions of old manuscripts were copied one from the other. This is Geoffrey Chaucer's Wife of Bath prologue in the 15th century, and there were many different copies of it. And you can use the same sort of techniques because the scribes would make copying errors, which are a little bit like mutations. And using those and these techniques, you can start to reconstruct the copying history. This is called stematology, psychology, people representing how we store our thoughts in our head, I guess, often use tree-like models. Whiskey, well, it's been used to classify single malt Scotch whiskey as well. And linguistics, well, there's the...
What's the variation?
Well, it's much debated. I think it's and of course, there's been a lot of... goes back a long way, the idea that languages evolve as well, like the Indo-European family, and some of the techniques that some of the speakers at our first meeting have been involved with have looked at more recent ways to develop this. So how's maths relevant to this question? I think there's sort of three areas. One is we can take the methods that biologists dream up and say, well, do they work? Often they describe an algorithm that they say does something, and sometimes it does and sometimes it doesn't, so you can sort of try and check, is the claim that it does what it says true? We can also try and develop better methods, and we can ask some fundamental questions like, is there really enough information in the data to draw robust conclusions? And I thought I'd do just three examples of the sorts of problems that can arise in phylogenetics, just to give you a flavour for this. The first is something called the Felsenstein zone. Joe Felsenstein is a statistician who in 1978 came up with this idea that suppose that this tree represents the true history of these four species, and one of the methods that biologists used in the early days of this field was just the following. You say, if two species somehow are genetically similar, then probably they are close together in the tree. So if imagine some sort of sequences or characteristics evolving down this tree, you'd expect A and B to be pretty similar, and C to be a bit different, and D to be quite different. So if you were building a tree by that process, you would infer this correct tree. You'd get the tree A, B, C, D. Okay? Because A would be closer. The data that this tree would produce would say A and B are sister taxa and C are the other. I've just drawn it as an unrooted tree because usually we represent relationships in this way. Now, Joe Felsenstein pointed out that if, however, the rate of mutation or the rate of evolution of the characteristics on this lineage here was much faster than everywhere else in the tree, you're going to run into problems because now these two things, A and C, are going to be the closest together. This is going to be quite different. So if you apply that naive method...
So the data is changing as you go along these lines, not just at the points where...
That's right. So we were saying somehow...
DNA is...
somehow here, I mean, we know that rates of evolution do change through time. Things suddenly start growing much bigger or whatever, or DNA starts going faster for whatever reason. And you'll see now that you're going to reconstruct the wrong tree, ACBD. And Felsenstein was able to quantify exactly when this would happen, what are the mathematical conditions for this to occur. For a long time, I guess biologists, well, some ignored it and some worried. But you can ask, well, does it happen in practice? And it seems that it does. There's been a number of studies since then. This is one example where John Hulsenbeck looked at different sorts of flies and genetic data, and there was this unusual grouping of Drosophila with this other type of fly. And when they analysed that further, they found that, in fact, it's probably an example of this phenomenon described, because you can now devise methods that will in some way recognise there's been a change of rate.
How do they do that if they don't have data today?
That's a good question. Let's just go back. If all you see is somehow how different are they, then in some way you can't. But you have all of the sequence information, you have all of the site patterns, you have the patterns that say, imagine you have two states, say red and blue, it might be eye colour or something. You see red, blue, 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 red, you have a whole lot of patterns for one observation, you have a whole lot for another. So all of that information together allows you to actually consistently reconstruct the tree, even when the rates change arbitrarily within it, if you have enough characters. If you just sort of blindly think of distances, count how many places are they different, then you can't do it. But if you take all of the sequence data...
But do you have to make assumptions like the rate of change is constant over the genome?
No. Yes, but then no. The no is that at first, yes, to do it easily, but if you have some, you can then generalize it to allow the rate to change across the genome, provided you have some knowledge of how that rate changes. If you have no knowledge, it does become much more interesting problem, and some people here are working on the question of really if you know nothing about this rate, can you recover it?
Is there any sense, is it always the case that you only have the final state information? If you're considering something like very small bacteria, do they evolve fast enough?
Oh, you do experiments. People do experiments.
Yeah. Can you verify some of
people have taken a population of bacteria and subdivided them, and then so you actually know what the tree is, and because they mutate or they choose a gene that mutates very quickly, they can actually simulate evolution, and they know what the underlying tree is. That's certainly one way. Another way is sometimes with the fossil record, now you can get DNA out of bones, for example, and you may have some sort of knowledge of these earlier times as well. That change of rate is one example of how evolution can be non-stationary, but there's other examples as well. In fact, one that caused a lot of worry in molecular biology in the early 90s was 18S ribosomal RNA, which seemed to, for some reason, put the birds over with the mammals, whereas they should be over in this group with the crocodilians. And it was realized that this particular part of the genome, what's happened is that the birds and the mammals independently had become rich in G and C. There are the four bases in DNA, A, C, G, and T, and they both independently become rich in G and C. And when you applied naive methods, they would group these together as though they were related, whereas you can design methods that would compensate for this, and they indeed separate them back into their right position. And the second one is what's called lineage sorting. This is illustrated in this figure, where you have three species that have evolved from some common ancestor. And suppose you take an individual here and you sequence a gene, and an individual here and an individual there, and you reconstruct the gene tree, the tree that describes the history of that gene. And what is this gene tree? Well, that individual got that gene from one of its two parents, and that parent got it from one of its two parents, etc. So you can trace back in time the history of that gene And somewhere in the past, these lineages will join up like so. And they can join up in many different ways. So this point here would represent an individual that had two offspring that carried that gene into these lineages here. But you'll notice something interesting about the way I've drawn this. If I just look at the gene tree here, it has a different topology to the species tree. So the species tree puts A and B together as assistive taxa, whereas over here we have over here we have B and C as assistive taxa. This tree here is really the tree ABC, just drawn slightly differently. So one can ask, well, how likely is this? I mean, clearly this is a problem if you're using genes to try and tell you something about species histories, if you can end up getting the wrong tree. And it turns out that for trees with just 3 leaves like this, the probability of this happening is less than the probability of getting the true tree. It's reassuring. Somehow it's saying if you put enough genes together and take the majority vote, you should be safe and sound, and that's what people are starting to do. But interestingly, when you go beyond 3 species, the story gets much more interesting. When you get to 4-leaf trees with 4 leaves, in certain cases, for certain shapes of trees and for certain assumptions about these population sizes, the most likely gene tree can be different to the species tree. And then something very remarkable was discovered last year, which was when you have 5 or more leaf species, you can always set things up so that the most likely gene tree is different to the species tree. Sorry to have drawn that. Sir John Kingman, I hope you'll forgive me. Yes, I was going to explain the coalescent at this point, but I think I mentioned it earlier. So this is this result here from Degnan and Rosenberg, who I think both are actually coming up to this program later.
Is there a most likely? Sorry, there's some rule here, like majority. Oh, is there another rule that solves this problem?
Yes. So this would rule out a simple majority solution, but it turns out that there is still a consistent way of doing this based on likelihood, for example, maximum likelihood or stochastic models.
So it sounds a bit like these theorems about the democratic voting rules.
Yes, yeah.
Where with enough candidates or situations, there's no perfect rule.
Right, yeah. Anyway, it seems an interesting result. Finally, so does it happen? Well, actually, we had a very interesting talk in our first workshop here on this lineage sorting issue with relation to the human-chimp-gorilla trichotomy. This has been actually a controversial question in biology for a number of years. It's somewhat been settled now, but I remember as a student it being kind of hotly debated. In the old days, I guess, 40 years ago or so, it was assumed that chimp and gorilla were a sister group and they'd split off from the lineage leading to human. But genetic data cast doubt on that, and there were a number of genes that seemed to be suggesting that human and chimpanzee were actually closely related and that gorilla had split off just a little bit earlier. But although that seemed to have been resolved, as more and more genes were looked at, some of them still started telling this other story, and it wasn't at all clear, but it seems now to be that lineage sorting is really the explanation for this, that this is probably the true species relationship, but these occasional genes that tell another story are due to lineage sorting. Finally, I'll just mention something about the problem of deep divergences. This is where you have some sort of a speciation event in a short period of time, and there's many of these things, such as the Cambrian explosion you may have heard of 550 million years ago. And then you have where there was a lot of speciation. Then you have long periods of sort of relatively little happens. And there's a lot of, it's often very hard to try and figure out, well, what happened there if a lot of time has passed, because any signal here has somehow been obscured by subsequent mutations. And this is very relevant to many problems in biology. One example is this, the origin of metazoans, or animals, if you like. You've got the arthropods, or crawly things. You've got the deuterosomes that include us, the mammals. So you've got, I need a pointer here. So you've got these animals here. You've got the roundworms and the flatworms and these little unicellular things, and you've got fungi over here. And there's a real debate in molecular biology about what happened way back here, hundreds of millions of years ago, in the origin of animals, really. Was there a split that put these together against the others, or was the split putting these two together, these two hypotheses that are debated? I won't explain this network. I think Daniel was going to talk about networks, but they basically display the conflicting signal here. So what do we know about these sorts of results? Well, some of the techniques, and actually some of these ideas come from statistical physics, allow us to set lower bounds on the sequence length. This is the sequence length you would need in order to figure out which of the three possible ways these lineages could be arranged. What's the sequence length you need as a function of the amount of time, if you like, of this speciation period, and L is the amount of time from here to the present. N is the number of sequences in each of these groups. So there's some sort of a trade-off. As you have more sequences or more species, it helps, but you've got this exponential factor that's working very hard against you. As the time goes on, it's sort of killing. Your sequences are going to have to get much longer. And this epsilon squared is if you halve this time, you somehow need 4 times as much sequence length. Finally, this is my last slide. In looking at the blackboards as we wander around, most of the time it's very clear whether it's the SIS group scribblings or the or our scribblings. But every now and then we look at it and think, is that one of our lot or one of their lot? So what are the sort of maths we use? Well, it tends to fall in, I guess, mostly into 3 categories. And mostly it's very different to the sort of math I think that you guys do, but primarily combinatorics, a fair bit of probability as well, and a certain amount of algebra and topology. This is becoming somehow more relevant to a lot of the things we do. And I think it's probably a good place for me to stop and hand over to Vince.
Okay, the next speaker is Vince Moulton from University of East Anglia.
So I'll talk a little bit about something to do with phylogenetic diversity. So when we were sitting down, Mike, Daniel and myself, thinking about what we could discuss, we felt it might be nice to try and choose a couple of topics now on diversity in networks, where people are sort of in the program are working hard on these problems, rather than to sort of just give a big spread of various different topics. So as Mike said, there are various types of mathematics being used to study the various problems in phylogenetics, and the type of mathematics that I'll be talking about are mainly falling in combinatorics, graph theory, and complexity theory. But before maybe discussing sort of that in a little bit more detail, I just wanted to give a kind of motivating problem. So this is a picture, sort of a part of a map of Australia, and people who work there have to make a sort of rather unpleasant decision in some sense. They have some water shortage problem, and they need to dam up some rivers to actually deal with this problem. And in the various valleys, of course, there are going to be different populations of various animals, and on one particular one they seem to be interested in are called spiny crayfish. And given that you have to basically decide which river you might dam up, you might start worrying about, based on, say, the fact that we're going to have to kill off essentially some species when we dam up various rivers, which particular river should we choose, okay? Now, of course, that's going to include very, very many different complicated social factors, etc., but in this particular study by Faith and Baker, they were looking at this particular species, and here they said, okay, we could either look at this particular river here that we might dam up, and then we would have to essentially dispense with D&D lineages, or we could choose, for example, this river, where we would choose A and E. So, in some sense, which is the best or the worst choice? So they made a tree, like Mike has been discussing. They took a particular gene from this crayfish, made a phylogenetic tree based on that gene, one gene, and then argued because if we basically take this, if we take this pair here, then we would have to dispense with lineage A, which gives this kind of length of tree or divergence, whereas if we looked at the other red place, we would have a sort of shorter amount of divergence in this tree. And based on this argument, they argue that essentially it would be better to, say, conserve any area essentially having A, because we're preserving more diversity. Okay, now, I mean, so there are a lot of assumptions we've made here. I mean, this is a complex problem, but people are essentially now, they are trying to involve more and more this kind of analysis where we look at the sort of genomes of the various things that we're having to make these decisions about, and use that as part of a factor in deciding which decisions to make. Okay, so that's actually a sort of fairly current problem that many people are involved in working on.
How computational complexity of reconstructing these trees go? Are these things NP complete?
Oh yeah, I'll discuss that a bit more. I mean, there are various methods to make trees. Okay, so there are sort of most of the methods I'll talk about are based on distances, which I'll discuss in a second. But there are other methods based on parsimony, or as Mike mentioned, likelihood. And depending on which kind of method you have, some are efficient but possibly less accurate, whereas things like likelihood are actually NP. Is NP complete to construct such trees, yet people find them maybe more appropriate. So it depends on, it's a rather sort of diverse field. There are many different ways, and it depends in the end which kind of problem you're working on and what you're willing to sacrifice, I feel, as to how long you're willing to wait for your computer, say, to spit out the answer.
Right, but if you're thinking about spin glass problems in stat mech or something, you might be happy with a ground state and an approximate ground state,
Mhm.
which would be like an approximate tree.
That's right, yeah,
The
that's right.
optimal ground state.
Absolutely, yeah. And anyway, even when you make an optimal tree, you have no guarantee that that's actually really
What happens?
it's the tree, yeah. So, I mean, maybe for a problem like this, possibly it could work out that even having a completely accurate tree isn't really sort of essential to make the sort of decision that you're interested in.
Presumably nature could have gone a different way.
That's right. I mean, as we've seen with Mike's talk, there are various confounding factors that could easily sort of make your method go wrong, as it were. So, so when we think about phylogenetic trees, if we're just thinking about it from this combinatorial perspective, we tend to think of them as graph theoretical trees, where we have certain nodes labeled. Here we have the labels A through G, and some edge weights, okay? And those edge weights are usually presumed to be somehow proportional to the amount of time, or the amount of change that has occurred over a certain period of time, okay? But if we're just now going to be think about them purely mathematically. No, we're not. Yes, if we. So yeah, I'm sorry to confuse you. this is what we would call an unrooted phylogenetic tree. You can also consider ones that have a root, where you indeed have a direction. Although, yeah. So yeah, so if I'm, at the moment I'm
why
just...
do you consider unrooted, I mean, since there is a direction?
That's a good question. I mean, we may simply want to understand... Sorry?
Most mathematical methods would not signify what was the root. This is why unrooted trees are mostly considered.
If you just look at the tree, at these nodes, well, they need not be able to.
Most mathematical methods would not guarantee what is the root, or just cannot tell what it was. The biologists can tell it by other methods like outgroup comparison. They look for something that they know to be entirely different, and where it joins the tree, it will give the root of the small tree.
Or at least some, approximation where it would be.
Most of the models that we use assume that evolution looks the same going forward or backward, and we think that's a biologically reasonable assumption. So therefore, the method can actually distinct. That is at the microscale process.
Yeah, but you're looking at changes, big changes.
Well, our data usually comes from processes that are at fairly microscale, like mutations of single DNA applications. And under those kind of conditions, we feel like things the process should look the same going forward or backward. I suppose physicists argue about this.
It sounds very much
Oh,
like
yeah.
the way physicists think about the second law of
No
thermodynamics.
CP violation in
Yeah,
nature.
that's right. So,
What are these numbers?
So these will just be some, I'm now basically going to just look at this mathematically. I'm just looking at these as some real numbers, okay? Some positive
'Cause
real
it
numbers.
comes from some metric on the
Yes,
space of these.
That's right. So I mean, at the moment I'm just sort of more or less divorcing myself from biology, to be fair. But yes, when you actually look at real sequence data, you'll take some sequences, and to get these kinds of distances, you will actually measure. You'll maybe count just the number of differences between the sequences, and that will give you some distance measure. So yeah, and so often what people will do is they'll come to you with a sort of distance matrix. On a set, and say, okay, can I represent, how should I now represent that by one of these trees? Okay,
But,
so
I mean, you're just given distances between
between the
those
leaves,
letters.
yeah. So they might come to you with a distance matrix on A through F or A through G, and then say, okay, here's the distance matrix. I've read it off from my DNA data. How can I represent it by such a tree? Can I represent
the information you'd be given that say that A and F would be distant by 4.5 units, and it's just a question of how to.
Yeah, so the first question you might ask, which is good that you've asked this question, is suppose I give you a distance matrix.
Yes.
How do I know whether it really can be put onto one of these trees or not? So if we look at this, we sort of notice that if you take any four leaves, that are labelled A, F, C, and D, and we look at this path here, if we add up this path and this path, well, that is actually the same. I'll try and use this thing. So the length of that path and that path is the same as the length of this plus the length of that. Yeah. So those two, for those four taxa, those two sums are equal, and they are greater than. Well, here we go. The sum of those two parts. Okay. So for any four taxa on such a tree, if you take these two, you'll have two of the sums equal and greater than the third. Okay. And indeed.
Non-Archimedean.
Ah, yes, that's right, yeah. And indeed, that essentially suffices to characterize when a distance matrix will indeed fit onto one of these trees or not. Okay, so that's sort of one of the sort of classical, as it were, results in phylogenetics. I mean, if you can think about phylogenetics, it's still a fairly new field, as you can see. So this is going back to, as Mike discussed, there were people already looking at problems in genetics in the 60s, and people were obviously have been considering sort of metrics in general for a long, long time. But This is one of the results that people are sort of use as a founding result again and again in the field.
Did you find connections with the toric geometry?
With
Toric geometry?
toric geometry. I mean, I know that Bernd-Sternfels indeed that there are some connections, yeah, with those kinds of fields. I mean, I'm not qualified to say what they are, but I've seen people who have been thinking about those directions, and I'm happy to show you those papers if I can dig them out. Yeah. So
this relation there also reminds of tropical geometry.
yes, that's exactly tropical geometry. I mean, we could discuss that for quite. I mean, that's something. Yes, also that Bernd-Sternfels and his group. I mean,
What is tropical?
it's essentially it's essentially where you replace things like plus and minus by max and min. Yeah?
David, it's what we call PQ webs.
And is the proof essentially trivial?
I don't believe that the proof is too strenuous. Let's say.
No,
It's not. Yeah, it's not.
you say yes.
Yes. It depends what you think trivial is, really.
Example sheet.
Yes, I don't know which proof they. I mean, in this proof here, I believe he did that by actually considering the structure of the tree in terms of things called in terms of the bipartitions that you get from cutting edges on the set. But I can't recall what it was off the top of my head. But. Or maybe Mike knows because you've written a book, so you probably had to. You probably have a proof there. Was that? Can you remember? Yes, that's right. Okay. So so back to the sort of in the spirit of the problem that we've been presented with the crayfish. Yeah, we sort of get a nice.
Sorry, just a question about the previous transparency. Did the theorem guarantee the uniqueness?
Yes, it does, and it's unique. Yeah. So, sort of in the spirit of that, this person, Dan Faith, started to define something called the phylogenetic diversity of a set of species relative to such a tree. Okay. Essentially, if you have a set. AF, CD, CB, and you want to sort of quantify the diversity of that set relative to that tree, he suggested that you should just simply take the total length of the tree spanned by those 4 species, and as you can imagine, then just as a little aside to give you a little flavour of how the field is progressing, you might ask, and essentially this is a question that you already just asked, suppose we had a distance on the set which really was satisfying that 4-point condition, then can we recover back the tree using, say, now PD measures? And indeed, if we're just given a phylogenetic, if we're just given every pair of elements we know the PD, that's just the length of the path between them. And as we've just discussed, you can then constructively get back the tree. That's something that's well known. Interestingly, if you now know the PD scores for every 3-element set, I mean, you can also, of course, get back the tree. But this really only sort of appeared fairly recently, in a sense, and indeed, it was only in 2004 that really people started to think about, just in general, can we get back a tree from all, say, M-element subsets. And so, I mean, I'm not trying to suggest that possibly these results are hugely surprising. I mean, they are nice proofs, of course, but the fact that somehow these things are quite recent, I think, just indicates that this is quite a new, sort of interesting field of research.
Is that square root of x? Or
No, that's the size of x. Yeah.
So x is
Yeah, that's the size of the leaf set. Yeah.
The A through G is X.
I think we've got a typo here to come in. I think that should be x plus the size of x plus 1 over 2, not the size of x plus a half. Sorry. Yeah. Sorry about that.
could be everything.
Yeah, that's right. It shouldn't be too hard if that was a half, that's right. It won't be possible, yeah, that's right. So,
best possible?
yes,
About best possible?
yes, they gave, they actually showed that, if you've got larger sizes, you can't do it anymore. So actually, that problem now, although that's been looked at 3 different times, it's now completely solved in a sense. So now we have an interesting, as a sort of, that was just a little aside, but now suppose we ask the following problem. Suppose now we have a phylogenetic tree. I'll try and draw it somewhere where I won't cover up. Suppose I've got some phylogenetic tree now, which I'm just thinking about as this labeled. It's got some weights on the branches, and I give you now, say, a number. Maybe I'll give you the number k equals 4, and I ask you, okay, can you tell me which 4 leaves I should choose so that the PD score of that set is the largest out of all possible sets of size 4? Okay. So for general K, I'm given this phylogenetic tree, and I want to know for fixed K what's the largest PD set. So as Mike mentioned, I mean, sometimes people people within the biological sciences will come across these problems naturally and then try and solve them themselves, and they won't necessarily prove that they're correct. And Dan Faith actually suggested the following approach, which I think you'll find is fairly natural. I mean, if you wanted the largest size 2 set, you would clearly just take the 2 leaves that were furthest apart, yeah? And then obviously the next leaf you would probably choose would be the one that was, so these are the furthest apart on the tree. The next leaf you would choose would be the one that, when you chose it, would contribute the most to the score that you have so far. So here you'll see that E indeed will contribute most to that score, and etc. So we could just keep adding in leaves in a greedy fashion so that our score always gets as large as possible for the next step. And the obvious question is, well, will this be guaranteed for any fixed K to give me a maximal set? And intriguingly, well, I mean, this is true, but interestingly, that was proven by Mike only in 2005, and also independently by Pardee and Goldman. Okay, so this is, I think, a nice example of what Mike mentioned, where you basically have algorithms in biology, and sometimes mathematics can come in and help actually sort of prove that that's the best that you can do, for example. So now, as you mentioned at the back, I mean, that's a nice answer, of course, because it's going to be very fast. So you might hope maybe in general we can just come up always with these nice fast algorithms for solving such problems. But suppose now, I mean, in general, when we're given data, we have a lot of actual factors. I mean, one of the big interesting problems in phylogenetics now is to actually start to combine not just genetic information, but to include, say, geographical information in analysis and various other factors. Obviously, when people used to build those trees, Haeckel built that tree back in the 1800s. He wasn't obviously having a set of DNA sequences. So how did he do it? Well, he used various characters, and now we have other kinds of characters like geographic distribution, etc. So suppose I'm given now, here we have some, I work quite a bit with a biologist in New Zealand, Pete Lockhart, who will come and join this program in November. He studies, one of the things he studies are New Zealand buttercups, and we could ask ourselves, well, if we made this tree now on, say, a set of 5 buttercups, and then we looked now at some area we could say, okay, what's the sort of diversity of buttercups in that area? Well, that will just be the sort of length of the tree spanned by the species that you find in the area. So the PD of an area, as it were, is just the PD score of the species that you find in that area relative to your fixed tree. And then you can go through the same kind of a problem where you have an optimization problem. You say, suppose I'm given a collection of areas, and you might think, well, this is seems a little bit abstract, but I mean, you might suppose some people are sitting in an office somewhere, they've got X million to spend, and they've got, say, to choose whether to spend that on saving this area or that area or another area as a nature reserve. Okay, how should we choose which areas? Suppose we've got a say fixed set of areas that we could select between, and we wanted to just choose some subset K of them to get the largest PD. Can we do that in a nice greedy way? And now the problems become no. Okay, it's actually now a hard. There is no efficient algorithm known, at least unless you believe P equals NP, for actually solving this problem. Okay, so although people who are here in this program now, Magnus Bordovich and Charles Semple, have shown that, well, at least if you employ some variance of greediness, you can get actually still fairly close to being optimal. And again, I think it's always a good question in the end. I mean, how important is it to get the optimal answer if you're actually rather more interested in the application? So that gives you a flavour and various problems like this can be then reformulated in terms where you might have some budgets and sort of how much things cost, and you can imagine that again all of those things will actually still be hard, but special cases can be done efficiently. Right, so that first analysis that I looked at on the first slide with the spiny crayfish, what they had looked at was a single gene. Okay, and as Mike said, though, we're now getting, we can actually now envisage not too far in the future where we'll be able to get whole genomes in possibly at least on small organisms, microorganisms in a day, and fairly cheaply. At that point, we're going to have now several thousand genes to consider. The problem of actually how to reconstruct phylogeny now, jumping from one single gene to several thousand genes, is in itself a whole interesting area of research that people are working on now. But suppose now I was faced with this in the context at least of the PD problem, I could say, okay, how should I take into account that now maybe I've got 6 genes to look at? And of course you could imagine there's all sorts of things that you could do. And one obvious thing that you could do is you could say, okay, I have now a collection of trees, all on the same taxa set, say. And it's kind of probably should. Okay, I have say 5 trees, all on A, B, C, D, E. Obviously, I could then define the phylogenetic diversity of a subset of that taxa just to be the sum of the phylogenetic diversities on each of those trees. So I could just take essentially the average PD over all of the trees. And then the question is, can we now find, again, the highest diversity set relative now to a collection of trees? And of course, for one tree, we know, yes, we can use greedy. And recently, together with a colleague of mine at University of East Anglia, we were able to show, well, okay, if you've got 3 or more trees, it's hard. And this left open the case, though, rather fun case for, well, what about 2 trees? Can we find an efficient algorithm or not? So we came to the program in beginning of September, and Mike wrote a little problem sheet up, and some people got working on that, and they have in fact solved that problem. And probably they should give that my part of the talk now, but I thought it would just be fun that you should see some of the things that people have been doing, say, here in the first month or so. they came with this solution. They said, okay, let's start with our two trees. Here we have two, both on the label sets A through G. And we sort of rotate them. And then what we'll do is we'll join up, say, G with G and F with F and E with E, etc. Okay, and get a new graph like this. Okay, and then they slightly modify that and put sort of some. They add in some extra edges. They put capacities on those edges with costs for using them. Essentially, the costs of the dark edges are just the length of the tree, negative the length of the branches, and then we have some. They have to add in some extra branches to make it all the sums work out. And essentially, they then show, yes, okay, if we look at this now as a network flow problem where we're trying to push k units from S to T, then say we fix k equals to 4, then we'll have to use, say, choose 4 of these edges here if we're forcing through 4 units. And that choice, if I choose any 4 here, then what's actually going to be, if I look back here, I'm essentially to get from here to here, I have to use this path. And say I choose this one, I have to use this path. So the sort of PD, so I'm basically got the PD score as being the cost. And the same, of course, on the other side by symmetry. Therefore, the sort of, to find the actual highest PD scoring set, you just basically then go ahead and solve, find the minimum cost, select minimum cost flow through that network. And of course, there are many ways known to do that efficiently. So essentially there we have a sort of an example of a pretty application where using sort of, I think, somehow fairly elementary techniques, but we have a nice solution to some rather well-posed mathematical problem at least. Okay, so I think that's all I had to say. So it's Daniel's turn now.
So any further questions about Vince's part of the talk? Sure.
We can always discuss over lunch because I think,
Okay, the final subject is Daniel Hewson from Tuning.
Thank you. The third part is going to give you a little bit of an introduction to phylogenetic networks. So I wonder what you think about this title of a paper. It came out in 2004. Mathematics is biology's new microscope, only better. Biology is mathematics' new physics, only better. So this is an essay discussing the importance of mathematics for biology and also emphasizing what biology might be able to do for mathematics. So there's a number of challenges posed in that paper. 5 challenges. Some of them were, for example, understanding the cell. We need new mathematics to be able to actually model what's going on in the cell, or understanding how a brain works. We need biologists are not going to be able to explain how a brain works without the appropriate mathematics that needs yet to be developed. And the number 3 in the list of challenges was replacing the idea of a tree of life, that all life is embedded in this evolutionary tree, by a network of life. And so this is really the following question that is discussed by biologists. Is this type of picture really a good representation of how evolution took place, or does it maybe look more like this? There are a number of mechanisms that biologists study where genetic information is not passed down vertically through time from parents to children, or from ancestors to descendants, but rather horizontally. And so some of these mechanisms are, for example, hybridization between plants. Sometimes hybrid plants, where you hybridize 2 plants, you bring together their genomes, this might produce a new species. Or horizontal gene transfer for bacteria, or recombination between different individuals, usually in a population. Let's talk briefly about horizontal gene transfer. So it's well known that bacteria, unrelated bacteria, can exchange genetic material through different mechanisms. One, for example, is conjugation, where the 2 cells come together, they open up this little tube, and then they can pass through what's called a plasmid, a small
chromosome which has some genes on it, and then that way they can swap useful genes. So bacteria species that's never been in contact with an antibiotic might become resistant to that just by spending time with other bacteria that already have a resistance gene. So there's a number of different mechanisms by which genes can get passed horizontally between different organisms, and the question is, do we really need to take that into account when we're trying to describe the evolution of life? And so I want to present a simple model of network, evolution along a network. So first, let me briefly point out a simple model of evolution along a tree. So in a tree, we usually think of in most simple models, we have an ancestral gene sequence here, and this is going to evolve through time along this tree. So you can think of the sequences that moving down through time here, and as they move along, they pick up mutations randomly according to some model, and then this continues further, and then at the leaves of the tree, you see these mutated versions of the original ancestral gene sequence. And the tree reconstruction problem, which is one of the main problems that people look at in phylogenetics, is we only see the sequences at the leaves of the tree. We have some model. We have some model by which they evolution by which they came about, and the task is to try and reconstruct the topology of the tree. Can we, just by looking at the leaves, figure out what the tree was?
So that must be totally dependent on what your model is, perhaps.
That's right. Yes. So the more general. So people are looking at the question. I mean, you need to be really general to actually model biology, but I think at some stage you just come to a point where it becomes impossible to, under the model, because it's so general to actually figure out what the tree was. So, yes. So usually the models that people assume are quite simple, and then everything works nicely, but then there's a problem. The simple model doesn't really match biology. And now, what would be a simple model of network of evolution along a network? Well, let's just change this tree into a network, and so we're looking at something like this now. And now here it makes more sense to, rather than consider an individual gene, maybe to think of a whole genome here. So here we have a genome, and we're going to see copies of this genome at the leaves of this network. And the question is, what happens here in this situation? And here we're going to think of the two genomes that exist at these two time points here, and basically what goes on here is there's going to be some mixture. So the sequence that arises here and goes down here is put together by a mixture, some part from P and some from Q. And now how this mixture actually takes place depends on the biological model. So if it's horizontal gene transfer, your resulting sequence is going to be mainly one with some small pieces from the other. If it's hybridization, then you can think of this just as a mixture. You've got some genes from the one and some from the other predecessor here. So, and now the evolution. Now let's look at the evolution of one particular gene. Let's say the gene is here, say. Now if you look at this gene, and now if you take the gene sequences up here, solve the tree reconstruction problem, then you're going to get this tree. Because in this particular example, we were pretending that the sequence was passed down through this ancestor. Now, if you look at some other gene, let's say gene number 2, let's pretend it's over here. Again, if we were to reconstruct the tree for this gene by looking at the sequences at the tips of the tree, we would get this topology. And so what we're, in this simple model of evolution, species, at some nodes you can take some of the genes from one predecessor or from one ancestor, and the others from the other. And so so we're making the assumption here that the history of any individual gene is still a tree. So the genes themselves, each tree, you look at any given gene, you get a tree. But these special nodes, they're called reticulation nodes, where the integer is 2. You might, because of these, you might see different tree topologies depending on which way the gene was passed down. So a network like this will give rise to 2 different gene trees. Sometimes the genes will be passed down from this ancestor, and in other cases it's going to come from here. And in general, if you've got k such nodes, then you can have 2 to the k different tree topologies. that arise. And so the game that biologists play is the following. They're given a set of gene trees, so they've sequenced different genes, they produce trees of those, and now they want to try and reconstruct a species network that gets rise to those genes, those different gene trees. So say in this example, you've got 2 gene trees, and you want to know how to fit these trees together, how to explain these 2 trees in terms of one of these networks. So that's what they like to look at different gene trees, and that they turn out, look at different gene trees that are incongruent. Can we explain the incongruence between the trees by some network in which they are embedded, or they were sampled from? Computationally, the problem is currently formulated in this way. You're given a set of phylogenetic trees. You want to find a network of the type that I showed you there, which has a property that it contains all those trees inside of it, and minimizes the number of those special reticulation nodes. The assumption here being that horizontal gene transfer and other reticulate mechanisms happen quite seldom, don't happen that often. And this problem, this has been shown to be an NP-hard problem just recently, and for 2 trees there's a fixed parameter tractable algorithm to solve that problem. So we've been looking at this over the last month or so to try and come up with a more general solution. And so I just want to give you a brief indication of what we've been doing here. So if you have a phylogenetic tree with these taxa A to F, then any node in that tree describes a cluster. It's just everybody who is above that node. And if you have a set of clusters, that someone gives you a set of clusters and you want to figure out whether they fit onto a tree in this way, then it's the concept of compatibility that plays a role here. And basically, the clusters have to be nested. One has to be, if you look at any 2 clusters, one has to be a subset of the other, or the other way around, or they have to be disjoint. So you have to have one of these 3 situations here. And it's obviously a trivial lemma that, well, it's a lemma at least, that if you take the Hasse diagram of a set of compatible clusters, then you get a tree. The Hasse diagram is a diagram that shows subset relationships between a set of sets. So you draw an edge between, so there's one node for each cluster, and you draw an edge from one to the other if the one contains the other, and there's nothing in between that's also contained in there. So if the clusters are compatible, it turns out to be a tree, the Hasse diagram. Now, so if you have multiple genes, you produce multiple gene trees, you take all the clusters, if they're all compatible, then you can just throw them together, you just get one tree that's going to be just a summary of the input tree. So the input trees basically would have to be the same, or one would have to be a resolution of the other. But the more interesting case is we have a set of clusters that are incompatible, so that they're not all nested with each other. For example, AB and BC, if you look at these, one is not contained in the other. AB is not contained in BC, or vice versa, and they're not disjoint either. They both contain the element B. And so what we usually, so they're joined by an edge. So if you think of these as vertices of node, the incompatibility graph would be you join two nodes by an edge if they're incompatible. And then you write down on this edge the one cluster minus the other, so in that case it would be A, the intersection of the 2 clusters, which is B here, or the other minus the one, which would be C here. Now, there's a couple more incompatibilities in this data set. It turns out that there's 3 pairs of clusters that are not compatible with each other.
So incompatibility is if all 3 of those sets are non-empty.
That's right. That's right, and then they're not nested. And now if we draw the Hasse diagram of this set of clusters here, then we get this diagram here. I've drawn this in small letters because this is not actually part of the input, but you always have the total set down here. And then you can see that all 5 of these clusters appear here somewhere. And you can see it's no longer a tree, and it looks something like that first network that I showed you. Okay. I guess I'll get closer.
But this is not a solution to the problem. This is not the kind of network that biologists are looking for. This would be too easy because it's, let me explain why this is not the kind of network that we were talking about briefly at the beginning. This is what we call a hardwired network in the following sense. If you look at one of these clusters here, it's hardwired in the sense that to find out who, so this node has this cluster here, and to find all the elements of this cluster, you just go down, you just follow the graph down to the leaves, and then you find exactly those elements. And here's another example for AB. You just follow the edges down to the leaves here. And this is not, should become apparent in a moment, that what biologists are really after what one might call soft-wired networks, which are slightly different. So we still want to represent these clusters, but in a slightly different way. So I'm going to take that edge out there, so I've just modified the network slightly. And now to see that all these clusters are still here, still represented, I have to move this cluster to somewhere else, because obviously if it's here, it's not, you can't reach all, the elements that belong to it are not below it or above it in this network. So I'll move this over here, and now let me show you that this network still represents these 5 clusters, but in a slightly different way. For example, ABC, it's still that cluster that you reach by going down to ABC, to the leaves. Now what about AB? The AB, that's the cluster I get by going down to A to B, but not going down to C. And this is the soft-wired aspect of this network, that whenever you have one of these nodes with more than one incoming edge, you're allowed to turn one of them off. And this corresponds to the fact that if you have multiple gene trees, that any given node like this, you might be getting your copy of the gene either from the one parent or from the other parent. And so in that sense, one of the networks that biologists are interested in are soft-wired. And here, so I've transformed this into a soft-wired network, and you can see that in this network there's only one of these special nodes, whereas earlier, before I did that little modification, if I get back there, there's 2. And so we get rid of that, and we get a network with exactly one reticulation node, one node where you're taking material from 2 different ancestors. And so we were playing with this, and then we noticed something here, that these triples of sets that you see here, that this is actually the key to figuring out how to find a soft-wired network that uses a minimum number of these reticulation nodes. And you can see here that whenever you have the incompatibility, that's not going to fit onto the tree, and you have to, in a way, get rid of all 3 of these incompatibilities. And the way to do it is to make one of the 3 sets empty, or to take care of one of the 3 sets in each of these incompatibilities. And the way to do it here is, you see that if I knew that C is a special optional taxa, then that takes care of this, because if it's allowed to drop out every now and then, because in a soft if it's connected by a soft edge, then you can get rid of this incompatibility. Up here you can get rid of this one, and you can also get rid of this one here. So the idea is, the algorithm is the following. You have a set of clusters that are incompatible with each other. And you write down all these incompatibility triplets here, and then you solve, and then you come up with the optimal solution by finding a minimum set of taxa that hits all these lines, in the sense that for each of these triplets, you've covered a whole one of the 3 sets. So this is a variant of what's known as a 3-hitting problem. And it's a hard problem to solve. But we already knew that this whole thing is a hard problem to solve. But once you've solved that, and you found an optimal solution, in this case it turns out that with these 5 taxa, which is minimal, you can take care of all the incompatibilities. We believe that we can show that you can always construct a network that also uses exactly 5 reticulation nodes. So that's a conjecture. And one of the nice things about working in this kind of area is that we do have people out there who are waiting for these results, in a sense. So we do turn them into software, and so that biologists can actually make use of the algorithms and apply them to data sets. And here's just one example of such a data set. So these are 4 to 6 alpine buttercups,
and two different gene trees, one based on a gene that comes from the nuclear genome and one from the chloroplast genome. And if you take these two together and you compute one of these minimal networks, then you see that there's a number of situations where there's something interesting going on. But there's only one that's of real biological interest. It's this one here, because you're joining up two positions in the tree whose last common ancestor node is way back here. And so this is a strong signal that the placement of this taxon here is due to, in this case, hybridization. And so if you've got something like this where the two ancestors are very near, or the lowest common ancestor of the two different paths of this node are quite near to where the node is, that could be due to noise, but a signal this strong is probably due to hybridization. And in fact, independently of this little analysis, it was already known that this is a hybrid plant. It's a hybrid of the two.
From some other variant.
Yeah, in this particular case, I don't know exactly how it works, but the upshot is that two plants, unrelated species, well, two different species, somehow throw their genomes together, and you get a hybrid plant who has twice as many chromosomes as has the chromosomes of the one plus the chromosomes of the other.
So is basically the idea is that's a very improbable event, that it happens.
It doesn't happen very often, but it does happen, and these plants manage to reproduce and form a new species. And then over time, people believe that quite often you have this plant has a high number of chromosomes because it comes from these two different species. But then over time, there's different mechanisms by which they can get rid of some of the chromosomes, get the number down to a more reasonable size. And so for us, the net result of hybridization is just a genome where you've got a mixture of genes, one from the one from the other.
What's the biological pressure that wants to get the number of chromosomes down?
I don't know about that. I don't know. Well, there's two things going on. For some reason, they do get rid of some of the chromosomes, but the other interesting thing is that you originally have in many cases you have two different versions of the same gene doing the same thing, and there's something called concerted evolution where the upshot is, or the result is, that the one copy tends to overwrite the other copy. So after a while, even though you had two different copies to begin with, they look the same because there's a mechanism by which the one gets overwritten by the other. Yeah, so that was just a little introduction to phylogenetic networks. Thank you.
The idea is we all proceed to lunch together as a group. So let's thank all the speakers for this morning.